Hello and welcome to the next Conversation with Giants in Medicine, hosted by the Journal of Clinical Investigation. I'm Ushma Neill. Today I have the pleasure of speaking with Stuart Kornfeld of the Washington University School of Medicine. Kornfeld is best known for his research in glycobiology, in particular, his work describing pathways involved in oligosaccharide biosynthesis, processing, and transport. These actions are critical in maintaining proper folding of proteins. Kornfeld also discovered how lysosomal enzymes are transported from where they are produced to where they're processed in the lysosome. As he was also an editor of the JCI in the late 70s and early 80s, I'm particularly interested in hearing about his life.
Well, it's a pleasure to be here.
Can you start by telling me a little bit about your parents and where you grew up?
Sure. I grew up in St. Louis, and I had wonderful parents. My father was a very fine dentist who had a private practice, but also was a professor at Washington University and St. Louis University. So he spent part of his time seeing patients, part of the time teaching and doing research in metals. My mother was his assistant. She ran his office and helped him at the chair. So they both worked quite a bit, but they were always there when I needed them.
And did they instill in you dentistry as the way forward?
No, to be honest, my father admired physicians. And I think always steered me toward medicine rather than dentistry.
So as a child, did you have aptitude for science and math and learning? Were you a good student?
I was a pretty good student, but no, my interests were really in sports. I lived in an area where there were a lot of kids, and this was before television. So we were always outside playing. And St. Louis is a big baseball town. And I was pretty good, so I got to play with the older boys. And I spent as much time as I could practicing so I could keep up with them. And science really wasn't on the radar till much later.
Well, clearly you were a good enough student that you got into Dartmouth. So what did you study when you went there?
Well, I was a pre-med. I was a zoology student, a major. So I took a lot of courses in the life sciences. But the course that really turned me on to science was a biochemistry course I took my senior year. And it just opened up a new world to me. And I was also impressed that in our textbook there was a large section dealing with the Corys, Carl, Carl and Gerty Cori, who were at Washington University Medical School. And it discussed the work they did on glycogen metabolism that led to the Nobel Prize. So taking that course and also knowing that I was going to be going to Washington University, I tried to get a job this summer before I could in the biochemistry department. And as luck would have it, my professor of biochemistry at Dartmouth had a good friend who was in the biochemistry department. So he contacted him and set it up. So I started doing research, real research, for the first time right before medical school.
So even though you were going to medical school, you started out doing research. So did you always have the thought that research was going to be part of your clinical training or maybe one of your focuses, or did you go into it thinking that you were going to be a clinician?
No, I always had the idea of doing medical research. But that was based on reading books about great discoveries in medicine. I mean, the book that impacted me the most, I think, was The Microbe Hunters by Paul de Kruif. It's a wonderful book, and everybody should read it. And this was my idea about research. It was rather vague. And I really had no idea about what you actually do in the laboratory. But I knew this was something that I was headed for. And then when I got exposed to biochemistry, this really showed me the way I wanted to go.
Now, did you do research all through medical school, or were you pretty dedicated into your clinical training?
Well, I was a good student. I worked hard. But I did research every opportunity I had. I worked in a lab with a mentor, Luis Glaser, and I was the only person there, so it was Louis and me. And he had just gotten his PhD and was just starting out on his own. So we were in one little room, and I would spend a lot of time with him, and he would show me how to do various techniques. So each summer I worked in the lab, and our senior year we had an elective period. I worked in the lab. So altogether, I spent about a year with Louis, and it was a great experience.
Did you ever meet the Corys?
Gerty Cori had died the year before I came, but Carl Cory's office was right next to Louis' lab. And yes, I met him, but he was very intimidating. And I was a medical student, and he was a Nobel Prize, world famous scientist. So he was very nice to me, but it was always a strain having to go talk to him.
Well, this is something that I think trainees would be interested in as well, is, you know, even if you are a medical school student or a graduate student or a fellow, should you go up and introduce yourself to these people?
Absolutely. Because once you do that, then you find out they're really very friendly and anxious to speak with you and learn about what your interests are, and it could be very helpful. Dr. Cori wrote a letter for me to get a position at the NIH, which at the time was very, very competitive. And I'm sure that had a lot to do with me being accepted there.
So after medical school, you did trundle off to the NIH for 2 years. Who were you working with?
I worked with another biochemist named Vic Ginsburg. And he was also very young. He had just come to the NIH. He worked in glycobiology. And it was a small lab. There was one other postdoc when we arrived. And when I say we arrived, I met my wife, Rosalind, at Washington U in the biochemistry department. She was a graduate student right across the hall. And when we went to the NIH, we decided we would both work in the same lab. We had common interest. So with Vic, it was Rosalind, me, and one other guy. So again, a very small lab group, but very interactive. And Vic was a very bright guy with a lot of interesting ideas, very helpful.
So for those of us who are not biochemists, why did you choose glycobiology? Because it seems a little bit confusing to me, quite honestly.
Really? Well, when I first started, my understanding of glycobiology really was the work the Corys did on glycogen. Glycogen is a polymer of glucose. And at that time, most of the emphasis was on energy metabolism and how glycogen is made and broken down. And the other area that was being studied was bacterial cell walls and yeast cell walls. In fact, my first project had to do with how yeast make mannan. But while I was working with Luis Glaser, I realized that mammalian cells also are coated with sugar moieties on their surface called the glycocalyx. And there wasn't much known about it at all. But people had seen it with various dyes and through electron microscopy. And the other thing I learned that really was important was that blood group determinants were composed of sugars, and that the difference between your blood group type being A or B is just a single sugar variant. And there were plant lectins, which are proteins found in the seeds of plants that could distinguish between blood type A and blood type B. So it seemed to me that these sugars on the surfaces of cells must have an important role. And there was relatively few people working in that area. So I thought that would be a great new area to explore once got on my own.
So you and Rosalind ended up back at WashU afterwards for your first faculty positions. So how did you go about setting up a joint lab or independent labs and determining who was going to do what? And what did you decide to focus your own lab on?
Yeah. Well, I should tell you that we got this position in a very different way than most people go about it today. We both had great admiration for Washington University and sense that it's a good place for young people to get started. And there was a wonderful chairman of medicine, Carl Moore, who I had trained with. And we liked St. Louis. So while we were at the NIH, we contacted Dr. Moore and asked, would he be interested in having us come back on the faculty? And he said yes. So, and the space that was available, well, I was interested in hematology, and they had lab space available for both of us. So when we first started, we both were interested in glycobiology. And we had some common projects. We also had, she had her own project and I had my own project. So we figured we would work together on certain things and go our separate ways on others, but always be in communication. It wasn't formally planned in any way, it just happened. And in the end, it worked out extremely well. If I had to name one research project that was most pivotal, it was a study that was done with a terrific MD PhD student in the lab, Ira Tabas. And it opened up a whole new area. So what it was is concerned with how lysosomal enzymes are tagged for delivery to lysosomes. And it was known from work of Elizabeth Neufeld that lysosomal enzymes have some sort of tag on them, on their sugar chains, I should say. Lysosomal enzymes are glycoproteins, which means they have covalently bound sugar chains linked to the protein. And Elizabeth Neufeld showed that there's a tag on these sugar chains that allows the enzymes to bind to receptors that transport them to the lysosome. But the tag was mysterious until Bill Sly, who was at Washington U at the time, showed that the tag is a modified mannose residue, a mannose residue with a phosphate on it. And this suggested that there must be a kinase that adds the phosphate. And a bunch of labs tried to find this kinase, and none were successful. So this is where Ira and I jumped in on this. We thought that if we could figure out in what stage in the synthesis of the lysosomal enzyme the tag is added, that we could then look in that area of the cell and find the enzyme that adds the tag. So Ira labeled cells with tritiated mannose, which gets incorporated in the lysosomal enzymes, and then isolated a lysosomal enzyme and first asked, does it have this tag on it? And sure enough, he was able to show, yes, this enzyme acquired the mannose 6-phosphate. And then as a control, to prove that it was mannose 6-phosphate, he treated the enzyme with a phosphatase, which Bill Sly had showed removed the phosphate. And to his surprise, the phosphatase did not remove the phosphate. My reaction was that Ira had screwed up the experiment somehow and used the wrong buffer, and that he should just go back and repeat the whole thing and do it again. But he did this, got the same result. And after puzzling over this for days, we finally got the idea that maybe the phosphate is blocked by another residue, and this started a hunt for what that residue could be, and it turned out to be another sugar called N-acetylglucosamine. And the beautiful thing about this was it immediately suggested an entire new biosynthetic pathway for how cells add a phosphate onto this sugar mannose. And then Marc Reitman and Ajit Varki joined the team, and in no time at all, we found a whole new enzyme pathway that accounts for how cells tag these lysosomal enzymes. And to prove this was the right thing, there were patients that Elizabeth Neufeld had described that can't add a tag to their lysosomal enzymes, and they secrete them into the blood. So their plasma has enormous levels of lysosomal enzymes, and their cells lack lysosomal enzymes. And Ajit and Marc Reitman showed that the defect in these patients is lack of this first enzyme that we had found. So the exciting thing about this experiment and how it just changed the whole way in which we think about how sugars are modified came out through a very careful study by Ira Tabas.
So I talked to Ira before this interview, and he told me a few things about you, that you have this very quiet demeanor. He also channeled your voice as we were talking. But he said that sometimes it was infuriating that you would come over and analyze the data before he got a chance to, but that it belied your incredible excitement about the findings that were about to come.
Well, yeah, I know. I've been accused of that. We've always worked as a group in the lab, and I communicate a lot with the people in the lab. I think that's one of the most satisfying things about working in a lab. My original impression of what happens in a lab is you go into a room all by yourself and do experiments and great things happen, and that's it. But in fact, there's enormous interaction among people in a lab, and that's very rewarding. There are many different ways you can mentor students, and they could all be successful. I just happen to have a style where I like to interact with each person in the lab. And when we've spent, some of these experiments take days and days and days to do, and the last step is when you put the samples in the radioactive counter And we're all very excited. So yes, occasionally I would rush in to look at the results, sometimes before the postdoc, but I try to avoid that most.
So let's talk a little bit more about clinical mentoring, because just as active as you were within the lab, you've also for many years shepherded the MD PhD students and also any clinician scientists that want to do any training. So why have you spent so much of your time on that topic?
Well, I was mentored by two wonderful physician scientists, Carl Moore, who was head of our department, and Saul Sherry. And they were both physician scientists who maintained clinical roles in the department and had done important medical research, basic research. And so this had a big impact on me. And I also feel that the clinical exposure is important for advancing medical research, and that physician scientists do have a special role. So I attended in medicine for many years and attended on our hematology service until just a couple of years ago. So I've had a lot of clinical experience myself. And I feel that the MD PhD program is the best way today for people to prepare themselves for careers as physician scientists. That they need the strong background in research training and also to have a good understanding of clinical medicine. And it's a difficult route, but it could be carried out. So instead of just talking about it, I thought I should be involved in these training programs. So I was head of the MD PhD program for 7 years, and then in the year 2000 we started a new program in the Department of Medicine called Physician Scientist Training Program, which is for graduates of MD PhD programs who are going to get further clinical training and also postdoc laboratory training. And it's a way of mentoring them and keeping their interest in medical research. And it's been very, very successful. We've recruited wonderful trainees to Washington University, and the vast majority of the ones who have finished the program are running independent labs and are serving as physician scientists, many of them on our own faculty.
You undersold your role a little bit. You weren't just active within the Division of Hematology. You ran the Division of Hematology as part of another scientific couple beyond the one with your wife. You and Phil Majerus together ran that division for many years and are often referred to as a unit of Stu and Phil. So what was that relationship like?
Well, Phil joined the department the same time that Rosalind and I came. And he had trained with Roy Vagelis in lipid biochemistry. And we hit it off. And we were both trained as biochemists, and we both felt that biochemical approaches to understanding normal physiology as well as disease, what was a way to advance medical research. And neither of us had a lot of training in hematology before we came. And Dr. Moore was willing to let us learn on the job to be clinical hematologists. But we both liked hematology. And then as it worked out, we did well and were promoted to be heads of the division at fairly young ages. And we worked, he had more interest in the business side. He took care of all the financial thing. I took care of the fellowship program, and together we recruited. And things went extremely well. We complemented each other. We were able to recruit quite a few outstanding faculty. Many of our trainees went on to be elected to the ASCI and the AAP. So it was just a very good collaborative operation for many, many years.
So, other than the 4 years at Dartmouth and 2 at NIH, you've spent pretty much your whole life in St. Louis.
Right.
So what is it about St. Louis, or WashU in particular, that's kept you so loyal?
Well, growing up in St. Louis, I really appreciate the town. I have a lot of friends, people even going back to high school who I still see all the time at the grocery store, things like that. St. Louis is a wonderful place to raise a family. There are a lot of terrific cultural opportunities in St. Louis, a wonderful symphony, some theater, enough theater. We have the St. Louis Cardinals, who I still follow very closely. And then there's Washington University, which is a great place for young people to start their careers. It gave Rosalind and me great opportunities, freedom to do whatever we wanted. Carl Moore felt that basic research was important for the advancement of medicine. So while he did not understand glycobiology, he felt that we should have the freedom to pursue this area, and he provided resources for us to get started.
But that was at the beginning. What has kept you there until now?
I just like the people there. I like the interactions that I have with basic people in the basic sciences. It's very easy to communicate with people. And we have terrific students who I've always enjoyed interacting with. And I still like living in St. Louis. So yeah, we had a number of opportunities to go to different schools, but decided in the end that none would be better than Washington University.
A couple of quick questions then. Do you recall your first experiment, even as a kid? Your first scientific experiment?
In college, I did a little experiment on feeding guppies with different amino acids. But my first real experiment, I must say, was with Luis Glaser. And that turns out to be a really interesting thing. So it was known that yeast have a cell wall. It's called the mannan. It's made up of mannose residues. But, and it was known that that was made from a nucleotide sugar, GDP mannose. So sugars get activated by being bound to nucleotide diphosphates. And then they serve as the donor for the sugar for making a polysaccharide. But it wasn't known how the GDP mannose was made. the Corys had found an enzyme that converts glucose 6-phosphate to glucose 1-phosphate. And we knew to make this mannan, you had to convert mannose 6-phosphate to mannose 1-phosphate. And it wasn't known whether it was the same enzyme that the Corys found or a separate enzyme. So my little project that summer, my first summer, was to see if one enzyme works on glucose 6-phosphate and mannose 6-phosphate are two different enzymes. So I had to fractionate a yeast extract and assay the fractions for these two enzyme activities. And I found that yeast have a separate enzyme. And what's so interesting about that, well, I loved the experience. And that's what really sold me, the final thing that sold me on staying in research and going through with biochemistry. But what's very interesting about that is in the 90s, a new class of diseases have been discovered called congenital disorders of glycosylation. Actually, the eye cell disease is the first one. But there are many more. And the most common one is a mutation in the enzyme that converts mannose 6-phosphate to mannose 1-phosphate. When I did that experiment right out of college, the last thing that ever occurred to me is that this work with ground up yeast would have anything to do with human disease. And yet it does. And without that work, they wouldn't have been able to make much sense out of the mutations that were found in these patients. So that was my first experience, and it shows in retrospect that doing basic work at some point in the future can have important clinical implications, and there are many, many examples of that.
Well, another recent example is your findings about stuttering and how some of these lysosomal enzymes and the changes here could be important for the pathogenesis of stuttering.
Yes, that is another remarkable thing. I was amazed when Dennis Draina at the NIH gave me a call and said he had been doing studies on families with persistent stuttering and found that there were mutations in the very enzymes that we had discovered that are involved in phosphorylating these mannose residues. And it's still mysterious as to how these mutations or how defects in this pathway cause stuttering, because the underlying neurologic basis for stuttering is still poorly understood. But this was the first genetic data on stuttering, and Dennis estimates that 5 to 10% of all people with persistent stuttering have mutations in this pathway. So, yeah, it's another example of you never know what's going to turn up.
If you could not be a physician or a scientist doing research, what sort of career path do you think you would have followed?
I think I would have tried to be an architect. When I was a teenager, I came across some articles about Frank Lloyd Wright and showing in a House Beautiful magazine. But it was displays of many of the houses he had done. And it just struck me that this was so beautiful. And then at Dartmouth, I took this course on art and architecture. And I really got to understand how architecture is developed. And there was a big section on Frank Lloyd Wright. So I've always admired him and that field. Now, I don't know if I would have the talent to do that. Certainly would never have been a Frank Lloyd Wright, but it's something I think would be very, very satisfying.
Well, I think speaking for the rest of us, we're all quite happy with the path you did choose. So thank you so much for joining me today to tell me a little bit more about you.
Well, thanks for having me.